Alright, so we're going to try to set up what's called our chromosome disease online lab or activity. And the first thing you need to do is Google the Karyotyping Activity. If you Google the word Karyotype Disease Lab, the first choice, the URL says biology.arizona. That's the right one. So you click here. So this lab activity is produced by the University of Arizona. Alright. First part of this is, well, it's called a karyotyping activity. That term karyotype just means a karyotype is a complete set of chromosomes for a person. And in laboratories all across the world, when they study problems with people, and you have a geneticist involved, and they're looking at their chromosomes to try to understand what might be wrong with a person, they look at all their chromosomes and kind of study and observe them. And when they get all the chromosomes set out to view under a microscope, it's called a karyotype. Again, that's explained right here. So the introduction of the karyotype, it shows you kind of what a real picture of chromosomes look like. So a lot of times in the laboratory when they get chromosomes out, they have these long shapes like this, and they have banding patterns. And the banding patterns and the overall length helps them kind of figure out which chromosome they're looking at. They get the banding pattern by applying certain dyes and chemicals to the chromosomes. They stain them. And the chromosomes always kind of stain or have certain patterns where they bond to the pigments or the chemicals in certain areas, and that creates that banding pattern that you see. All right, not all chromosomes are the same length, and not all have the same banding pattern, so when you look here, you can kind of tell that. That's an important detail because you're going to have to create karyotypes in order to better understand someone's disease. So when you're kind of going to be researching certain patients to figure out what's wrong with them, and you'll use the way their chromosomes look like to try to figure out what's going on. All right, let's keep going down. Right here is your assignment. It says, this exercise is designed as an introduction to genetic studies on humans. And again, it says karyotyping is one of the many techniques that allows us to look for several thousand possible genetic diseases in humans. It says you will evaluate 3 patients' case histories. So you'll learn about the patients. It says you'll complete their karyotypes and diagnose any missing or extra chromosomes. Once you identify which chromosomes they're missing or which they have extra copies of, you'll then conduct research on the internet to find websites that cover some aspects of human genetics. If this is an assignment for a class, you should turn it in a total of 7 answers on a paper. All right, we'll talk about that soon, don't worry. Here is a copy of the worksheet that you'll be working on to complete. This worksheet, if it's in digital form, of course has a link here and it gets you there faster. And it has a little guidance on how to get there. But anyhow, there's 3 sections to this worksheet, and each section is one of the patients. You start with patient A, then you'll do patient B, and then patient C. It says, what is patient A's history? So you summarize what's the life history, what's the past, or what do we know about this patient, right? And that could be their complaints, their physical problems, or, you know, just about their parents or the individual themselves, right? Any information that's important about that person. And that's on the website. So let me show you how that works. So you get patient history by clicking the patient history button, of course. And right here, it says patient A. This is their history. So what you're going to do is read this statement about their patient history. It says patient A is the nearly full-term fetus of a 40-year-old female. So patient A really is a baby that hasn't been born yet. It's inside a 40-year-old woman, and it'll be born soon. It says chromosomes were obtained from the baby from fetal epithelial cells acquired through amniocentesis. So they were able to go within the mom and get to the tissue of the baby and collect cells. In your own words, summarize this in 1 sentence, and you do it on the worksheet. So right here, you would summarize patient A. Who is patient A? You'd write it right there. In your own words, summarize it, okay? All right. Now your next topic is, it says the last set of chromosomes is the sex chromosomes. If you have 2 large chromosomes, your patient, if you have 2 large chromosomes, your patient is XX. 1 large and 1 small indicates that they're XY. What is the sex chromosomes that your patient has? So you got to figure out the gender of your patient. So right now we can look at patient A's chromosomes, and let's check it out. Now, these are two chromosomes of the same size, and these are small chromosomes, so these would be considered rather large. So this person is XX because they have two large chromosomes here. So we'd call patient A, that the baby that's yet to be born, it's going to be a female. So you'd put XX, and you'd say that it's a female. It says which chromosome set has an extra, so which chromosome is there extras of? So I'm going to do this first one. I'm going to do patient A with you, but your responsibility would be to do patient B and C on your own. So let's go back and see if we can't figure this out. So which chromosome do they have an extra copy of? So I'm looking at this chromosome right here. And what your job is it says place this chromosome in the partially completed karyotype. This is the karyotype. You can see all of this person's chromosomes are set up. But just like in a lab, you have to kind of sort them and put them in the right place. You have to match. And just like you have two of each allele or trait, you have two of each chromosome in your body. So we have to complete the karyotype. So we have to figure out, where does this chromosome go? Where is its partner? So we have to figure out, is this chromosome 1, 2, 3, yada, yada. Now, you can see that 2, 5, 12, and 16 are highlighted. They're in blue. So in this example, because I'm a genius, I can tell that this chromosome here matches best to chromosome number 2. And so it looks like they have 2 of chromosome 2. And it says correct. Now click on the chromosome in the karyotype that is homologous to this chromosome. So I got to figure out which one does that match. I'm going to say 16. Another correct match. Click where this chromosome belongs. So I'm going to say that one belongs with 12. And then this final one will be 5. It says, now that you've paired up all the chromosomes, where does this extra chromosome belong? So patient A has one too many of one of these chromosomes. And your choices are 18, 13, or 21. I think this chromosome looks most like 21, so I'm going to click 21 because I think they're a match. It says, congratulations. You've successfully completed patient A's karyotype. All right. Now that we've finished the karyotype, we realize that the patient has one extra copy of chromosome number 18. So the baby's only supposed to have 2 of chromosome 18, but it actually has 3 copies of chromosome 18. And so that's really the problem. And if you have an extra 18, you pretty much got a disease. So you'd put right here in this blank, you'd have an extra copy of chromosome number 18. And it says, what diagnosis would you give this patient? What's the disease? Let's see if we can find that from the website. All right, it says making a diagnosis. The next step is to figure out what disease they have based on having an extra copy of 18. And in this example, it looks like, let's see here. Oh, it wasn't an extra copy of 18. Sorry, that was my bad. So it has an extra copy of chromosome 21. So this baby would have Down syndrome. So we change this. We'd say that it wasn't chromosome 18. It was actually, they had an extra chromosome 21. And we'd put right here that they have Down syndrome. All right, now repeat the steps for patient B and C on your own. So you go to patient B by just clicking patient B. All right, good luck.